Good afternoon, ladies and gentlemen, and thank you for coming to our first of the series of lectures as part of the 2015 CCOVI lecture series. Today we have with us our first speaker is Vincenzo De Luca. He is a professor of biological sciences and plant biochemistry here at Brock University and also a CCOVI fellow. He's also a Tier 1 Canada Research Chair in Plant Technology. His educational achievements or degrees include a Bachelor of Science in Honours, Bachelor in Science Honours degree, in Biology from Concordia University in Montreal, Quebec, a Masters of Science in Plant Biochemistry from Queen's University in Kingston, Ontario, and a PhD in Plant Biochemistry from Concordia University in Montreal, Quebec. His research is focused on functional genomics, and particularly secondary metabolism in grapevines and in various medicinal plants as well, such as the Madagascar periwinkle. He seeks to improve wine quality parameters and to enhance production of valuable medicinal products. He also teaches plant growth and development and topics in plant and microbial biotechnology, current topics in basic and applied plant biology, and mechanisms in plant biochemistry. So without further ado, ladies and gentlemen, please step forward, Vincenzo. Please welcome, a warm welcome to Professor Vincenzo De Luca.
Thanks very much for your kind introduction. I'm happy to be here today to tell you a little bit about small efforts that we've been making to try to look for easy ways to monitor the appassimento process in grapevines. As you can imagine, this is proving to be rather difficult. Easy things are always seem to be always difficult to achieve. I'm going to talk mostly about the work that Dr. Kyung-hee Kim in my lab has been doing over the last several years. She's been involved in the appassimento project that has been financed by the Ontario government within CCOVI and with other researchers around Ontario. I'll also touch a little bit about some work that we've initiated and we're planning to continue with Kimberly Kathleen, who's an MSc student who also works at the Vineland Research and Innovation Center. And this other work is a part of a collaboration with Dr. Bernard Goyet, who's over there. So if we look at the appassimento process, you can actually produce grapes that are being dried under controlled or uncontrolled conditions. This will affect grape metabolism through the modification of some of the chemistries. It may modify the types of volatile aroma compounds and other organic molecules that are produced. And this includes a range of polyphenols, the various fertaric acids, things like caftaric acid, the flavonoids that you find in grapes, as well as chemicals that appear to be antimicrobial that are produced often in response to fungal infections or fungal interactions. This is things like resveratrols. In addition to that, there are a range of anthocyanin compounds that are there and molecules such as tannins that may be modulated as a result of the appassimento process. The whole process involves dehydration, and this is normally monitored by people producing these grapes by monitoring weight changes over the process involving water loss. Grapes usually have to be sacrificed in order to monitor things like sugar concentration. And usually appassimento grapes are harvested or processed for making wine when they reach a certain sugar content that has been predetermined by the grape, the winemaker, for example. These methods are more costly, they're more labor intensive, and they are time consuming to do. And unfortunately, they're not always effective. There's been complaints about the relative quality of some of these appassimento wines that have been produced lately, as more and more of the wines have been produced around the planet. One of the things that we are really interested in is how do we develop an effective and reproducible process that will give us rapid, accurate, and broadly applicable technologies that can be used by winemakers. The appassimento process itself leads specifically to increases in the sugar concentration, and you'll find that often wines made from these types of grapes have higher alcohol content. And they concentrate other substances, which is really through the elimination of water. And these other substances are really what give the particular cachet of these wines and involve increases in aromatic compounds, their color, the phenolic substrates in the must. And these are processed by the action of catalysts called enzymes. The other thing that happens is some of these aromatic compounds are converted into polymers, and they become more complex in their structure. And much of the trick is to identify what are the components that are actually relevant to producing fine appassimento wines. Some of the things that happen during the process is the disappearance of the so-called rustic grassiness, some of the bitterness, and producing more of a smooth and rounded type of tannin as a result of the complexity that's generated. The other thing that's really difficult to quantify is the effect of things like desirable botrytis effects or some of the undesirable gray molds that chemical changes that take place that you really don't want to have. And I think these are areas that are of active interest to producers of appassimento wines and not so easy to determine the right approach to direct the grape towards one or the other. The process leads to more changes in chemistry, glycerin, the breakdown of chemicals such as malic and tartaric acid, and the degradation of certain varietal aromas, similar to things like production of various acids and volatile colloids, and very importantly, the action of catalysts that lead to breakdown or modification of the chemical entities. And these catalysts may, in fact, not necessarily come from the grape. They may be coming from microorganisms that are on the surface of the grape. And this is very difficult to determine what the main causes of the changes in the chemistry that are taking place. So today I'd like to focus on a couple of topics. First of all, the appassimento project that we were involved in with other colleagues within CCOVI and within the community at large and other researchers. And many of you probably realize that the focus of the project was to study the appassimento process when grapes are maintained on the vine under barn conditions, in the greenhouse conditions, within a very controlled drying chamber, or in tobacco kilns. And I think various wine producers are using one or more of these approaches to try to develop fine appassimento processes. I'll talk a little bit about some work that's going on in Italy over here, where there's a much more elaborate set of experiments were done involving transcriptomics, proteomics, and metabolic approaches to try to find markers. And as you'll see from that project, it's a huge amount of money that was spent to try to find markers using that approach. And I think you'll find at the end of this that it's not been so easy to find appropriate markers. There's lots of markers there that are now identified as possible components, but which ones are the right ones to use for producing high-quality appassimento? In my own presentation, I'd like to talk a little bit about what happens in ice wine grapes, since some very interesting results were obtained a few years ago by us in terms of some of the biochemical changes that take place when grapes are left on the vine till the point comes for harvest and for making ice wine. And I'll talk about the fact that we've been monitoring a whole range of polyphenols and trying to see if their patterns change as we go towards appassimento. And it'll range from things like simple phenols to various resveratrols, some cyanidins, flavonoids. You see there's a whole range of flavonoids that are found within the grapes, and anthocyanins. If we look at the production of ice wine grapes, it's really interesting that as you look at the level of resveratrol, for example, in ice wine grapes during the development of ice wine grapes, you'll notice that the level of resveratrol rises from the month of August till later on in December, and then sometimes even into the end of December. And it's interesting that we looked at several varieties of grapes, and in all the cases, we found that resveratrol levels rose, and they rise something like 5 to 10-fold. And that's pretty interesting because the grapes are already ripe at that point in the month of August, September, and the resveratrol levels keep rising. And this is here shows you both resveratrol and pisiid, which is a glucoside of resveratrol. And what we see is that these levels are rising, and it may be the fact, due to the fact that there are fungal contaminations during the month of October and November, for example, and these may be the ones that are involved in somehow activating the biosynthetic processes that lead to resveratrol biosynthesis. So one of the questions we had when we saw this type of data was, does this involve de novo biosynthesis of resveratrol? Are they made from scratch, for example? So one of the things we did, we actually looked at a gene called stilbene synthase, which is the protein product of that gene is involved in the actual biosynthesis of resveratrol. And you have to produce the precursors, of course, because you need things like paracumaril coenzyme A, as well as malonyl-CoA, in order to allow that particular reaction to take place. So this particular data set shows that both the transcript levels are increasing here as you go towards later stages of development, later on during the month of October and November, and it correlates with the increase of resveratrol content within these grapes. So it's telling us that not only is the gene for stilbene synthase expressed, it must mean that the metabolites required for the making of resveratrol have also to be made, because CoA esters are not simple things to make. And that to us was very interesting, and we've been putting together a patent relating to this because it could be of use to us in terms of harvesting tissues that make high levels of resveratrol. Okay, so one of the questions that this led to when the appassimento project came was what are the metabolite changes that might be taking place during appassimento grape production? And as you know, these things are staying on the vine and they're exposed to the elements. Appassimento is grown, are grapes that are harvested for the most part, apart from the vine experiment, and placed under different control conditions. So to make a long story short is, we looked at resveratrol levels and we asked, well, what about other metabolites within these ice wine grapes? And we did not see any changes in anthocyanin levels, for example, really. And if we looked at things like furtaric acids, we did not see lots of changes of furtaric acids. So the whole thing seems to be restricted to resveratrol. And that may be, again, due to the fact that resveratrol is a phytoalexin that is activated by fungal pressure. And that part of it would need to be verified, of course. So if we now go to the metabolites that are being produced during the appassimento process, this slide shows you measurements that were done in vine, barn, kiln, greenhouse, and drying chamber. I'm sorry that the curve is smooth here. There should be just a series of points showing you the data set. But one thing that you notice is there's a fair bit of variability in the levels that we're measuring from one time point to another. And all we can do is look at the pattern, the general pattern of change as illustrated by that straight line that we see over there. And one thing that you'll notice when you look at the measurement of things like gallic acid and caphteric acid, we notice that under the different treatments, and this is at the end of when we harvest the grapes for actually making wine, you'll notice that the final levels of things like gallic acid don't change a heck of a lot, but the level of caphteric acid has risen to some extent under all of the treatments compared to the grapes that were harvested at the outset. Okay, so we're seeing a 3- to 4-fold increase in caphteric acid, and of course, one of the things that's been happening is that they're losing water, and therefore you have to take into account the water loss versus the final amounts that are there, and whether there is really more synthesis or simply a concentrating effect that we see over here. Just to make it easy, I've given you the structures below of caphteric acid and of gallic acid over here. Now, if we go to look at still beans, and this is in 2013 over here, once again, you'll notice that the still bean levels, are they really rising? You'll notice here the control, and we look at individual still beans. So I've got what the cis and trans resveratrol are down here. And you'll notice that the other things that are found are pisiids. The pisiids are glucosides, so it's just resveratrol with a sugar added to it. And finally, in blue, we'll see the total resveratrol content, which is simply an addition of free and bound forms of resveratrol. And you'll notice that there's the levels. I mean, does this mean there's an increase? Really not terribly, except for this dry, the controlled drying process that has higher levels. And to me, this does not parallel the kinds of things that we saw when we cultivate the grapes, the ice wine grapes, for example, where we actually did see, and we did see this reproducibly over several years, where the resveratrol levels were rising. So, leaving these under these in various types of growth conditions here, or controlled growth, temperature conditions, you don't really see the results that we saw with the ice wine grapes. And if you now take this over a 3-year period, you'll notice that the levels do vary, first of all, from year to year. And we had discussed this a couple of years ago when the 2012 resveratrol content was very low. You'll notice here that the maximum levels that we see are about 8 micrograms per gram fresh weight in this case, the barn version. And in 2011, the numbers were closer to 30 and 35 to 40 in 2013. So this is really a reflection probably on the disease pressure that the grapes are undergoing. So resveratrol content is really a great marker for the amount of fungal contamination or disease pressure that the grapes are undergoing. So one of the interesting questions here is, are these chemicals produced more when you get a gray mold or when you've got botrytis? And this type of thing should be done probably under controlled conditions to try to verify if resveratrol could be a good marker for the appropriate type of molding as opposed to the inappropriate type of molding. And one of the most frustrating things about doing this type of work is that you can only do it once a year. And that part of it makes it very difficult to do this type of experimentation because you can't do it over again. You know, you have to wait till next year, till the next grape harvest is coming in. And unfortunately, there's no alternative to this for the moment. So we went back and we looked at catechin levels. And under each of the growing conditions in 2013, and again, you notice that there really isn't a huge change from control, right? We're seeing variation here, and it could simply be variation. One thing I forgot to mention about resveratrol, if you notice here, the drying chamber had significant amounts from the previous slide, but here you notice it's got lower levels. So it's not like this tells us, well, the drying chamber is going to give us higher levels of resveratrol. How many times do you have to repeat it before you get enough statistical information to tell you that this is a relevant result or not? Okay, so the catechin levels have not really changed in each of the treatments. Now, we go to flavonoids, and you can look at, in general, at flavonoids, or you can look at specific flavonoids by monitoring the levels of each one. So if we look at a very well-known flavonoid known as quercetin, it exists associated to various sugars or acids over here. So glucuronide, glucoside, and this is a methylated form of quercetin, which is also a glucoside. And if we look at the patterns over here, once again, we notice that the quercetin glucoside is the most abundant form with small amounts of the others. And, is this, again, not a tremendous change in the pattern that we see? And one of the questions you have to ask yourself is are we seeing a shift towards other forms of quercetins that might be more stable, for example? Because if you play with the sugars that substitute the flavonoid, or you start to decorate these in any way, they may become more stable. Okay? And is that a component in quality? Once again, I don't have an answer for you there. But so if we look at quercetins, not a heck of a lot changing. If we look at camphorol, the difference between quercetin and camphorol is it has one less hydroxyl group. Okay? Same biosynthetic pathway, but simply no hydroxylation at this position here. And again, we see camphorol glucosides and galactosides over here. And we notice once again that the glucoside is the major camphorol derivative. And once again, really not a heck of a lot of change. We're up to 4 to 6 micrograms, whereas here we've got 200 micrograms. So you know that the major flavonoid of grapes is quercetin. not camphorol. However, when you're doing appassimento, you have to ask yourself, is there a shift that might be taking place? So if you look at the individual flavonoids, so far we don't have a heck of a lot of change taking place in terms of the types of flavonoids that are there. So here we've got anthocyanins now, and when we look at the profile, I mean, it doesn't take a lot for you to say, well, not much is changing, right? So these are total anthocyanins in 2013. And if we now look at specific anthocyanins, and these are all malvidins, and malvidins are anthocyanins that have methyl groups on the B ring over here. And you've got glucosides, you've got glucuronides, and you've got glucosides, and then you've got an acylated anthocyanin over here with this acyl group, and then you've got a paracumaril associated anthocyanin over here. And once again, if we look at the major anthocyanins, you notice that they are the malvidin glucosides. And here, of course, what you're seeing is a general increase in malvidin glucosides, as well as the acetylated forms, and a little bit more of this more complicated aromatic associated form over here. So this is simply drying, and the anthocyanin levels are rising in a fairly consistent pattern over here. And when I was looking at the Italian work, they were claiming that they saw increased levels of these acylated forms over here within the appassimento grape. And that's very interesting, because as you start to modify the groups at the end of these things, these anthocyanins become more stable. Their stability increases, and the Italian groups claim, well, it's because the levels of these are rising that we're producing a more colorful product. And, you know, I'm not sure that these slight increases in these acylated forms make the difference. And, of course, they don't tell you in detail within their papers the levels. All they do is, principal component analysis, which is there's lots of statistics that you can use to tweak the system in one way. This is raw data, and it shows you that the changes are not really as important as they might otherwise be. So if we now look at the 3 years, you'll notice that the anthocyanin levels are, I mean, there's some change over here, but they're not that different, once again. So this result tells you one thing, that the level of the ratios of these anthocyanins is different, but overall the levels are the same. So there has been a change in ratios. What's the consequences? What does that mean in terms of what the final product looks like? And of course, in the end, which of these treatments produces the better wine? That's really the aim of this whole work. And so far, the metabolites that we see aren't really telling us much about the final product that's generated by the different drying treatments. And that's very frustrating, because we do see changes, but we're not able to correlate that with quality. All right. So I went back to this Italian work, and it was kind of interesting. There were 13 authors on this work, and it was just a massive piece of work. As you can see from the title, Identification of Putative Stage-Specific Grapevine Berry Biomarkers and Omics Data Integration into Networks. And so this involves a lot of different skill sets. First of all, as you can see here, they're going to do transcriptomics. So I took this out of Wikipedia there, So what's transcriptome? The set of all RNA molecules, including messenger RNA, ribosomal RNA, tRNA, and other non-coding RNA transcribed in one cell or a population of cells. So this is, the RNA is the component of the cell that produces it's a transcript that will then be used in making protein, right? And the protein is the business end of the cell, which actually does catalysis. That's involved in making metabolites, for example. And so to do a transcriptome, in 2010, what they were doing were microarrays. So a microarray is basically a chip on which you've got 30,000 genes imprinted. And then you can actually take those 30,000 gene chips and monitor which genes are expressed, all right? And so you can say, okay, at this stage of development, I've got, 9,500 genes that we see out of the 30,000, and 6,000 are upregulated, 1,000 stay the same, and the other 2,000 are downregulated. And then you can infer that these genes are involved in this or that part of the process during the developmental stages. And these researchers took berries at different stages of development over here. So you can see highlighted what PV stands for: pre-veraison, veraison, pre-ripening, ripening, withering 1, 2, and 3. So you have each of these stages of withering that's taking place, and they've harvested RNA from each stage, and then they use that to do a microarray. Okay, and then you get all these genes that you see that are up, down, or staying the same, and they now integrate that into a profile. So, how do you do that? Well, you need the person that's going to do the chip technology, and then all those genes have to be put into a database, and you need bioinformaticians who are going to be able to take all that and assign them to different functions or different genes that are already, because the genome of grape is already known. So the next component was to look at proteomes. So now here what you do is you look at the entire set of proteins that are expressed within that cell or that tissue or that organism. And it focuses on each of the stages that the researchers looked at within this cartoon over here. And the same thing was done with the metabolome. So this refers to the complete set of small molecule chemicals found within a biological sample, a tissue, or an organism. So if you look at a particular cell, how many metabolites are you going to find within a cell, right? The best labs in the world are able to monitor, let's say, up to between 500 and 1,000 metabolites. That doesn't mean they're able to measure precisely how much they are there, but they can detect that by using, sophisticated mass spectrometry methods. And but there are probably, 3,000 or 4,000 metabolites in a cell. Some of them are in very, very low quantities. So you can see this is technically speaking, very difficult to do each of these components. So these folks use microarray technology to do the RNA work. Today, you would use RNA-seq. So that is basically harvesting the RNA and then doing large-scale sequencing. Today, RNA-seq will give you 50 million sequences in one round of sequencing. So you could actually take each of these time points over here and sequence each stage of development. You get 50 million sequences. If you want to do it 10 times, you now have 500 million sequences. And then you can ask, how reproducible is those sequences that you see? And it'll give you an idea of which transcripts are being expressed at each stage of development. The same thing can be done with the proteins. You harvest proteins, and then you can run gels where you separate proteins in 2 dimensions. The first one is based on what is known as the isoelectric point of the protein, and they'll separate according to their charges. And the second dimension is according to molecular weight. And the best labs in the world are able to separate 10,000 proteins using this type of method. So if you have 10,000 proteins on a gel, the proteins then can be transferred to a solid membrane, and each protein spot can be harvested. And you can take that spot and digest it with proteases and then send it to MS analysis over here. Okay, and because we know the genes and the proteins that they encode within grapes, this MS will say this protein is found at this stage of development. And so you know, very powerful techniques, but you can imagine the workload to actually do this. It's an enormous task, and one would expect that what they've done is they look at the 2D proteins and see if the level of a protein goes up or down. You can actually see it by staining that its level has changed, and then you can say, well, I'm going to sequence that protein, because if you do it with 10,000, what would the cost be? So this here, RNA-seq costs about $1,000 for one round of sequencing today. So if one was to say do 10 of these, that would be 10,000, 20, 30, 40, 50, 60, $70,000 to get 500 million sequences for each time point. Okay? This is incredibly inexpensive. In 2003, to do 60,000 sequences was $60,000. Today you get 50 million sequences for $1,000. Hence why, you know, we're going to be getting people asking us in a few years, would you like to have your child sequenced when they're born, right? Because they're going to be able to predict what sicknesses that child is going to have. And of course, I have no idea if we'll be forced to do that, or whether we'll have the right to say yes or no. And all the ethical issues that raises, of course. So this is a very, very powerful technique, and this is one of the things that we'd like to do in this project that we're working with Kimberly on in the next year. Finally, the final part of it was metabolome, and this involves high-performance liquid chromatography combined with mass spectrometry, and as sophisticated as that sounds, you really don't have I mean, it's hard to do lots of metabolite analyses, and you need lots of standards, and it's not a trivial exercise, and it's fairly expensive to do. Okay, so actually the cheapest part of it is the RNA-seq, as long as you have a good bioinformatician around who can help you with the organization of all that data into something that's usable. So you have all this happening, and then the researchers will do these network organizations. The details aren't important. You can always go look at the paper, but they do transcript protein network, they do transcript metabolite network, and then they do protein metabolite network. And that gives you a way to bring everything together to give you a transcript protein metabolite network. So now you can associate the genes that are expressed to the proteins that are made to the metabolites that accumulate. Okay? And one thing that's fascinating is this gives you a massive amount of information. And here I've highlighted what each of the symbols mean. This is the transcript, protein, and metabolite. Now, what this illustrates is parts of biosynthetic pathways that have been modulated or changed. And you can see here, you've got genes that regulate transcription. So these are transcription factors, for example, that will activate the production of a particular messenger RNA and will in turn increase the production of that protein. And you can see that a whole bunch of genes stress, withering stress responses, for example. So oxidative stress, biotic stimulus. So these were things that are activated by microbial attack or fungal attack, or things like loss of water, dehydration, and so on and so forth. And what's interesting, it's giving you a bunch of targets. And when you read this paper, you realize that they've got targets, but they're unable to associate it with quality, right? So how do you go from this data to having a useful tool that a winemaker can make to say, this is the time when I should be harvesting my grape, okay? So here we've got a bunch of proteins that we can look at as targets that we might want to see if we can use those in order to equate it with quality. The other one is the phenols. I mean, everybody focuses on phenols because we know how important they are in the quality of the wine. And so they show, for example, branches of the biosynthetic pathways, that this is a common branch leading to phenylpropanoids, things like caffeic, caftaric acid. There is resveratrols over here, and the anthocyanins down here. And one of the things that you notice is that acylated anthocyanins seem to have gone up, which was what I was telling you in the previous slide, as well as some of these resveratrol-related metabolites, the viniferins, which are dimers of resveratrol or trimers of resveratrol, and this glucoside of resveratrol called pisiate. So with this kind of data set, you say, okay, how can I now turn around and develop a tool that I can use to monitor the withering process and find the right time for giving the advice saying it's time to harvest, to make your wine now, okay? So there's a lot of data. Now the question is, how can we find potential markers for quality? And one can actually look at transcripts. It's very easy to do, RT-PCR and quantitate the level of a particular transcript, and one can see this type of tool could be in any winemaker's lab because it's becoming very straightforward to do these types of things where you can monitor the level of a given transcript. It's pretty inexpensive to do that, but maybe a protein marker would be even better, right? And you could actually check all the markers that I showed you in the previous slide and ask if you could make, for example, an antibody reaction where you can actually monitor the presence of the antibody. And if it's a single entity, it would be simple and inexpensive. Imagine a dipstick method that all you do is you crush your grape and use your dipstick, and it'll tell you it's time to make wine. The problem is it's not likely to be simple, right? It may be much more complicated, and it might involve multiple entities. And this would be more costly and time-consuming. The other one that you can use are, again, small molecules, but you have to be able to say which small molecule. So far, we have not been able to identify appropriate small molecules. The best small molecule is sucrose still, right? The sugar content. And we would like to have other molecules that we could use to actually monitor that. And so my position is that we may be able to find appropriate entities by simply testing a series of these proteins to see which ones of them might be best suited for monitoring the quality. And there's an awful lot of data out there that's available for us to actually go to that stage. Because if we do more of this kind of work, we're not likely to, we're getting big picture, but we're not getting into the details. And I think that's where I'm finished. Yeah. Thank you very much.
Thank you, Vincenzo. We do have some time, a few minutes for some questions if anybody has any they would like to ask.
Okay, just to finish off, this is a... Oh, sorry, I didn't see you there, Steve.
I was going to ask, actually, of these markers, I wonder, are there better candidates that will survive through to final wine, and that perhaps then you can work backwards from quality to these markers in the final wine, and then use that as a selection method for the markers to then be using in the grapes as they're drying? Like, are there ones that perhaps do correlate better to final wine chemical analysis?
Yeah, well, if we're talking about metabolites, we've been looking at a lot of those metabolites that you see in the final wine, and the problem is that we're unable to differentiate between the drying treatments. I mean, the whole idea was if we found one drying treatment that is preferential over the others, why is it preferential? And could we assign a metabolite that would give you that information? And so far, the analyses have not yielded specific molecules. We clearly know that resveratrol is a very good marker for disease. So I think if we can show that we're getting a botrytis effect and that produces a better wine, we could look at resveratrol, for example, but it's very tenuous, right? It's not as cut and dry as it might otherwise be. On the protein side, most of the proteins disappear once the final product is made, although some of these osmotic type osmotins and so on will be found in the final wine. Again, my sense is we need to be able to correlate the protein to the product that's made. And, one could go back and look at the final wine, but there's, very small broken down things maybe that are still soluble, but not the original. So, yeah, we could look at that. I don't know if people, I think, have looked a little bit to peptides. Peptide sequencing is relatively straightforward today. One might go back there, and I'm not sure if in the literature if people have done that to any extent. I know they've worked on peptides, but I need to look more carefully what's going on there.
Could I ask you just briefly to expand a little on what you were just saying just now about resveratrol being a marker for disease in grapes?
So what we noticed is that when the disease pressure is higher, we see higher resveratrol levels within the grape. As far as we know, resveratrol is made in cells in the skin. There's very little evidence that resveratrol is made in other parts of the grape. It's not made in the seed. It's not made in the pulp. It's all skin specific. And there's been a lot of work that has demonstrated that. For example, if you take grapes that are growing under, are ripening, and you expose them to higher light intensities, you'll actually see higher resveratrol levels. And that has nothing to do with disease. That's a light treatment that triggers resveratrol production. If you treat, you may have temperature effects too that will activate resveratrol production. But in many instances, disease itself triggers resveratrol production. We do not know in the ice grapes that we were looking at what level of disease was in there, but we saw reproducibly over 3 years that resveratrol levels rose between August and November. If you wait much longer, they start to break down. Now, does that mean they've broken down or they've been leached out? We don't know the answer to that question. But you can actually infect the grape with an organism and you'll trigger resveratrol biosynthesis. So that's why I was saying it might be a useful, I mean, I don't know how we would go to that next step. That's where the trick is.
Okay. Wonderful. Any other questions from the audience? No, thank you. Thank you very much, Vincenzo. A little gift from us. And just to remind everybody watching that next week we have Wendy McFadden-Smith, who's an adjunct professor of biological sciences, a CCOVI professional affiliate, that's right. And her topic is viruses, vectors and vines, oh my. That's her words. That's her words, not mine. Thank you very much, Vincenzo.